My name is Ben Liesfeld. I'm a Managing Director of Limbus Medical Technologies, and I'd like to take the opportunity and give you a brief overview of how we support high-performance NGS liquid biopsy assays, and at the same time help our customers to be compliant with IVDR. First of all, who are we as a company? We are a software development company, a medical device manufacturer at the same time, located in northern Germany. And our products are cloud-based clinical decision support systems. Our main software, Varvis, has been a CE-labeled medical device since 2015. And we focus on NGS data processing. If you take the entire workflow of next generation sequencing, you can divide it into two parts. One part on the left is performed in the wet lab, and the other half on the right is basically data processing. And Varvis covers all the steps, starting from the raw data from the sequencing instrument to the report generation. As I said, the software is a cloud-native software and a medical device. That means there's no investment in IT infrastructure required. We can cater to customers all across the world with the same level of service. And our software scales nicely from small focused assays up to the largest assays like whole genome sequencing. And sometimes liquid biopsy assays can also be considered very compute-intensive assays. We are deeply rooted in human genetics as a company and as our product as such. And we aggregate information from all of our customers into a centralized data network. This network today consists of more than 94,000 data sets. And we also, these data sets also comprise family history. We have clinical information about the patients. So far, we gathered information about 37 million genomic variants, biomarkers, if you will. And we are particularly proud that our software has been used to diagnose 3,800 distinct diseases so far. And we would like to create the same benefit for even a larger number of patients using NGS for liquid biopsy. In order to explain a little bit the properties of NGS assays in liquid biopsy, I'd like to compare it to the other main technology used in liquid biopsy, ddPCR. If you look at the number of biomarkers that you can detect in a single assay, ddPCR probably gives you the ability to detect about 25 biomarkers in a single assay. With an NGS assay, you're able to query 25,000 distinct nucleotides. Not all of them may be significant for disease, but the assay behaves, or it needs to be considered like performing 25,000 tests at the same time. Regarding the limit of detection, ddPCR is considered slightly more sensitive than an NGS assay, but both these parameters, the number of biomarkers that you can detect as well as the limit of detection, it's not so much a technical limitation of NGS, it's rather costs that limit your the breadth of the assay. And this is an example of assays, NGS assays, as they are employed in care today. If you're looking at the key challenges that those two technologies pose, as I said, digital droplet PCR only delivers a few biomarkers, so the use case is rather limited, or it's tailored towards cases where you know what you're looking for, a well-defined collection of biomarkers. NGS, on the other hand, poses quite different challenges. The data processing becomes a challenge, and if you're thinking about performing 25,000 tests at the same time, the positive predictive value of each test is important. You need to be able to reduce the number of false positives in your results. And as other speakers in the morning have highlighted, there are at the moment very few IVDR compliant commercial tests for liquid biopsy on the market. So most of our customers are making use of Article 5 and IVDR. They are developing their own laboratory developed tests, in-house tests. But that means that these health institutions now become a kind of manufacturer in the sense of IVDR. And that means they need to demonstrate that they comply with the general safety and performance requirements, and they need to comply with many other requirements. So they're almost like a regular manufacturer. But most importantly, the general safety and performance requirements tell you need to evaluate the performance of your assay very well. And this is where we help our customers. In general, there are the two very important parameters that you need to determine are sensitivity and the positive predictive value. You need to be sure that your result is correct. And ideally, you have a reference material that gives you both a large number of variants of biomarkers that you can verify, but also a low background noise across all the other markers that are contained in your assay. And in our experience, we have seen issues with several reference materials from various manufacturers, which will make it almost impossible to perform a good performance evaluation and to characterize your assay well enough. I think one of the next speakers will be one of those manufacturers who actually can deliver good reference material, because if you don't have suitable reference material, the challenge becomes much bigger, because then the health institutions need to resort to custom samples or need to derive material from patient material and need to apply custom protocols. Also things that we've done in the past, but this is not the ideal solution. And in the end, I'd like to highlight two examples that I consider groundbreaking diagnostics from two of our customers. One of them is the Immuno-Oncology Center in Cologne. They're using NGS liquid biopsy to monitor treatment progress of their patients. They use it to detect resistance against therapy as quickly as possible. And they also use it to detect new molecular targets in the course of the therapy. And the other use case I would like to highlight is quite different from cancer diagnostics. It is rare disease diagnostics, and this is from the University of Magdeburg Institute of Human Genetics. And they are using liquid biopsy to diagnose patients with rare diseases, but not regular rare diseases, but even more complex mosaic diseases, where pathogenic variants are not present in all cells in the body of the patient, but only in certain organs or certain parts of the body. And liquid biopsy in these cases is really the only approach how you can properly diagnose the disease. And the Magdeburg therefore really caters to patients with great need, and we're really happy to support them in that. And if you have any further questions, please visit our website, varvis.com, or contact us directly. Thank you.